So welcome to my biology revision series. Especially if you're sitting for GC exam, this may prove to be really helpful for you. So I'm actually revising the 2023 question paper, that is internal biology 2023. So let's go through together. It says figure 1 shows changes which take place when a plant cell is placed in a solution of non-concentration non-concentration, that is from stage 1 to stage 3. That's what they tell you there. Then the question is, so you can see the diagram, stage 1, 2 and 3. So you can see they have labeled. So this is basically a plant cell. You can see the change that has actually happened, that the cell membrane has started detaching itself from the cell wall, and eventually, because of losing more water, it actually further pulls away, and you can see what is happening to the middle part called the vacuole. It is actually shrinking because water is actually being lost from the cell. So the cell keeps on losing water. So as it keeps on losing water, it simply means that the part which we are calling, so this part that you are able to see here is simply the cell membrane. So the cell membrane has actually detached itself there. So basically that is what is happening there. Then it says identify the parts of the cells labeled A and B. So what is A is representing the cyto, and that is the cytoplasm. And what is B there representing? That is representing the vacuole. So that is what you needed to write in the exam. Then you're good to go. Then the outer part that you are able to see here is what we call as being the cell wall. Then of course this part that you are able to see there, that is representing the nucleus. And this part, like I said, that has detached away from the cell wall, that is what we call as being the cell. So that is the cell membrane that you are able to see. So you should be able to identify the parts even as you are revising. It will help you in the exam, because some of the parts they have not asked are parts that they can ask you later on. Now notice B, it says B1 says using the changes shown in figure 1, explain what would have been the concentration of the solution. So what would have been the concentration of the solution in which the cell was placed compared to the cell sap. So compared to the cell sap, the solution that is found in the vacuole, the cell sap. So what would have been the concentration? So basically we could have actually said that it was actually the solution was more concentrated. So we could have said the solution was more concentrated. So it was actually concentrated. Now there's a word, if you want, that you could have actually used when we say it was actually more concentrated. Obviously you could have said the solution was, in this case you could have used the word hypertonic. So it was placed in an hypertonic solution because it's a solution whose concentration is higher than that of the cell sap. I hope that is actually helping you. Then the other question there says, what term is given to the process that led to the changes shown in stage 1 in figure 1 from stage 1 to 3. So remember, because this was placed in a cell and it was losing water, so what term is given to the process that led to the changes? Yeah, the process that led to the changes, it's because of a process we are calling osmosis, the movement of water molecules. So water molecules were actually moving out from the cell, and water molecules will move from where they are in a high water potential to a region where they are in a low water potential. So because this cell was surrounded by a more concentrated solution which had less water, water will start moving from the cell going into that solution. So the process is called osmosis. Then C says, what would be the overall effect of this process on a plant? So what would be the overall, if this process continues, if it is a plant continues to lose water in its cells, then it would actually lead to what we call wilting. So the overall effect is that the plant would wilt. Yeah, so what we simply call, in other words, wilting. Yeah, the sagging of delicate, parts of a plant in this case. So wilting would have actually, started taking place, and that is what you needed to. Yeah, so the plant would actually wilt. Yeah, losing water. No wonder you see certain plants, if you have not given them water, they will start sagging. It's because of this process. That's what we call wilting. So wilting can either be permanent or it can be temporal. Where a plant cannot be revived, then that's permanent. If it is temporal, then you can just water the plant and the plant will be revived. That's the way it is. Look at D. It says, which of the 3 stages of the cell in figure 1 would be classified as turgid state? So which of the stages, or no doubt about it, turgid is where the cell is, bulging or swelling. It's basically, we could have said stage 1. So stage 1 was actually the best there that we could have actually highlighted. Then the other one says, name 2 other processes which can make substances to move into and out of the cell. So remember when you did transport, across the cell membrane, the other ones that we can simply talk about apart from that is what we call as being diffusion. So that's another process. So diffusion, which is actually a form of passive what? Passive transport. So passive transport does not require, the use of energy. So diffusion is an example of passive transport. Even osmosis is an example of passive transport because they do not require, energy for them to move. They are both moving from a region of high concentration to a region of low concentration. But for for osmosis, it involves water molecules moving from a region of high to low across a selectively, or a partially permeable membrane. Then the other one that we can simply talk about is what we call as being active water, active transport. So as opposed to passive transport, diffusion and osmosis, active transport requires energy for you to move materials because you are moving against the concentration gradient. So these are 2 other processes that would have actually, taken place. I want you to look at, let's come to question number 2 and see how you would have actually answered, this question. Number 2 was saying figure 2 shows processes of the fate of glucose, produced during photosynthesis. The letters C to I represent these processes. So you can see, remember when the process of photosynthesis takes place, it simply means carbon dioxide is combining with H2O. Of course, there are conditions that are there. You need sunlight in the presence of chlorophyll that helps to trap that sunlight. Then you produce food C6H12, then O6, but at the same time you are going to produce oxygen. So oxygen is actually going to be produced. But our interest is simply the glucose, and that's what you can see there. So the glucose is actually going to